Hi, welcome to the Med School Phys podcast. My name is Greg Rodden and I'll be your host. I am a third year medical student now. I just took my first step of boards and I think they went really well. So let's talk about what we're doing here at Med School Phys. The idea behind this podcast was to try and fill a gap that I'd noticed in the podcasting world. There really aren't that many people talking about medical physiology and pathophysiology at a level that's pertinent to medical students. So I wanted to try and fill that gap. I love to teach. I love physiology and I love podcasts. I consume a lot of media through that format. So I figured why not blend the three together and try and make a product that people can both learn from and enjoy at the same time. Now, let's talk about how I plan to structure each episode. So what I'd like to do is I'd like to start with some announcements or a preamble, then dive into the content of each episode. For example, this episode is going to be about muscle contraction. And when we're moving through the content, I'd like to use a Socratic method, basically just asking questions as we're moving through the material to try and engage my listener a little bit more. I found this to be very helpful for other students and for myself, especially in the first 2 years of medical school. So we'll see if that works well for people. And then after we move through the content of the episode, I'd like to do a couple of board-style review questions at the end of each episode for both that are relevant to both Step 1 and Step 2. And hopefully people will find this method fruitful and enjoyable at the same time. I have never done anything like this before. I've never even recorded a YouTube video before, so I have a sharp learning curve ahead of me. But my sincere hope is to constantly improve and to put out content in the podcast format that is both academically rigorous and hopefully fun to listen to at the same time. The nice thing about listening to podcasts is that you can listen to them on demand and you can do them on your own time. For example, I listen to them a lot when I go walking and when I'm driving to hopefully maximize my productivity in things that would otherwise be kind of downtime. So, with that introduction, let's get started with the episode. This first episode is going to be focused on skeletal muscle contraction. So to start with, what is the purpose of muscle contraction? Well, it's to move our skeleton so that we can interact with our environment, right? Now, one of those physiology questions that they love to ask is, what is the fundamental functional unit of a skeletal muscle? And the answer to that question is the sarcomere. So the sarcomere is made up of these important proteins. Can you name them? Well, the two main ones are myosin and actin. So which one is thick and which one is thin? Myosin is thick and actin is thin. But we've got to know a little bit more than just which one is thick and which one is thin, right? So we know that on actin, there are these myosin binding sites, and those myosin binding sites are important because they allow the myosin to grip onto actin and pull on actin so that we can ultimately shorten the sarcomere itself. Additionally, there are proteins that help to regulate the interaction between actin and myosin. Can you think of their names? Troponin and tropomyosin. So troponin, that sounds familiar, right? We think about troponin when we think of a heart attack and some of the labs that we'll be drawing. And tropomyosin, so why is tropomyosin important? Well, tropomyosin helps to regulate the interaction between actin and myosin by actually blocking up those sites on actin that myosin will bind onto. And so how do we get those out of the way? How do we get the tropomyosin out of the way of the myosin head? Well, we have troponin in the mix. So there are different types of troponin. but one of these troponins will actually bind calcium, right? And calcium is released upon muscle stimulation by a nerve. So do you all remember which troponin actually interacts with calcium? It's troponin C. So when troponin C interacts with calcium, it ultimately causes this conformational change in the whole complex, and moves tropomyosin out of the way, and that ultimately allows myosin to bind to actin. And of course, this is only in the when we have ATP available. So the next topic is to talk about the organization of a muscle. So we know that the sarcomere is the fundamental unit of contraction. However, groups of sarcomeres ultimately form what? Myofibrils. And then myofibrils form into groups within what? Myofibers, right? And myofibers are the individual muscle cells themselves. And then muscle cells organize themselves into fascicles. And then fascicles will ultimately organize themselves into a full muscle. So surrounding these structures that we just listed, we have a few important structures called the endomysium, perimysium, and epimysium. So endomysium surrounds what? Surrounds individual muscle fibers. Perimysium surrounds the muscle fascicles, and then the epimysium surrounds the entire muscle itself. And one important thing to remember about the perimysium is that something courses within the perimysium. Well, it's actually the blood vessels and nerves that help to supply the muscle. So the next topic that I'd like to cover is the contraction cycle itself. The contraction cycle can seem a little counterintuitive at first, so we're going to take it step by step. And the place that I usually like to start is thinking about what happens when ATP binds to that myosin head. So this is what you've got to keep straight. When ATP binds to the myosin head, that actually causes relaxation of the muscle. It does not cause contraction of the muscle, which is something that I always seem to confuse. Okay, now ATP has attached to this myosin head, causing muscle what? Relaxation. Okay. ATP is attached, and then eventually myosin has this function where it actually hydrolyzes the ATP. The myosin head can also be referred to as a myosin ATPase. Okay, now we have myosin, ADP, and phosphate all in a complex together. Okay, and when the ATP is hydrolyzed, it's the actual hydrolysis of ATP that will cock the head of the myosin such that it's in a position that allows it to do work. And that makes sense given what we know about ATP, right? ATP is this high energy molecule, and when we hydrolyze it, we're releasing the energy stored within the ATP to allow the muscle to do work. All right, so now we're in this position where the ADP phosphate myosin complex is cocked and ready to do work. Then what happens is calcium has to come into the mix, right? And we said that calcium binds to troponin C. And when calcium binds to troponin C, that moves tropomyosin out of the way, allowing the myosin ADP phosphate complex to bind to actin itself, right? So once that complex has bound to actin, the phosphate will be released, right? And when the phosphate is released, that causes another conformational change in this complex. And the conformational change that occurs is one that releases the energy stored in the cocked head, right? And that's the actual pulling motion of myosin on actin. So one way that I like to think about it is like you're cocking the hammer of a gun, right? So when you're pulling the hammer back, that is the action of the ATP that was hydrolyzed by the myosin head. And then calcium comes in and binds to troponin C, and then that moves tropomyosin out of the way. So calcium is like moving the bullet into the chamber. And then when the phosphate is finally removed from this complex, the trigger is pulled and the bullet exits the gun, or the muscle contracts. All right, so I hope that analogy was helpful for some of you. And the next topic that we're going to dive into is the neuromuscular junction and the events that lead up to the actual release of calcium that facilitates skeletal muscle contraction. So the neuromuscular junction is one of those things that sounds scary, but it's really not that bad when you get into the details of it, or at least into the details of what you need to know for your board exams. So there are several important components to remember when thinking about the neuromuscular junction. So when the action potential reaches the axonal bouton at the end of the axon of the nerve, and again, this is the nervous side of the neuromuscular junction, it causes the influx of a certain substance, and that substance is calcium. So when calcium enters the cell, it causes a cascade of events to ultimately release a certain substance known as acetylcholine. And acetylcholine is released from vesicles, and there are certain proteins that facilitate the release of these vesicles, or the release of the contents of these vesicles. And those proteins are known as snare proteins. So those are the major things that you need to remember about the nervous side of the neuromuscular junction. On the muscular side of the neuromuscular junction, probably the main thing that you need to remember is the kind of receptor that the acetylcholine will end up binding to. Do you remember what that receptor is? It's the nicotinic receptor, and they can try and trip you up with by putting down both nicotinic and muscarinic as answer choices, but in the case of skeletal muscle, it is the nicotinic receptor that is stimulated by acetylcholine. Okay, so now we have acetylcholine bound to its receptor, and acetylcholine binding basically causes depolarization of the sarcolemma, or the plasma membrane of the muscle cell. And depolarization of the sarcolemma is carried into this system of folds of the plasma membrane called the T-tubule system. So these T-tubules are folding in towards the center of the muscle cell. And on either side of the T-tubule, you have portions of the sarcoplasmic reticulum, which is basically the endoplasmic reticulum of the muscle cell. And an important substance is stored in this sarcoplasmic reticulum, if you remember what that is, and that is calcium. So the calcium that's stored here needs to be released. And the way that it's released is the action potential that is traveling down this T-tubule will ultimately stimulate a receptor called the dihydropyridine receptor. And that receptor is physically coupled to another receptor. If you remember what that's called, it's called the ryanodine receptor. So the ryanodine receptor is actually a calcium channel that helps to release the stored calcium within the sarcoplasmic reticulum into the cytoplasm of the muscle cell to allow calcium to do its work on troponin C and facilitate muscle contraction. Okay, so we've covered a lot of ground We've talked about a lot of different aspects of muscle anatomy and physiology. And some people might be wondering to themselves, okay, why in the world are we talking about all of this detail and proteins and yada yada? And the answer is because each and every one of these components can become damaged or defective and cause pathology. So let's go through a couple important examples. One example that we didn't quite cover here is the example of Duchenne's muscular dystrophy. So I'll bet that most of you can tell me which protein is defective in Duchenne's. It is dystrophin. And dystrophin is important, but why is dystrophin important? Dystrophin is this protein that helps to physically link the sarcomere to the extracellular matrix, right? We have these sarcomeres that are contracting, but if they're inside of the cell, how in the world are they actually transmitting force? And it's because the dystrophin, along with several other proteins in a complex, is helping to transmit that force to the outside of the cell and then ultimately through a tendon and then ultimately to move a bone. So, if that's the case, then you would expect there to be profound muscle weakness in Duchenne muscular dystrophy. And there is, but there's also many other symptoms related to progressive muscle degeneration. So the next example that we ought to cover is myasthenia gravis. So myasthenia gravis, as most people know, is an autoimmune disease, and it's important to know what is being targeted in this specific autoimmune disease. And the answer to that is the nicotinic acetylcholine receptor. So we have these autoantibodies against the acetylcholine receptor, and this is causing the various symptoms of myasthenia gravis, symptoms such as ptosis and double vision and fatigue that gets worse throughout the day. So naturally, the next disease to cover is Lambert-Eaton, and Lambert-Eaton is similar to myasthenia gravis in that it's an autoimmune disease, but its autoimmune target is on the other side of the neuromuscular junction. And do you remember what it's targeting? It's targeting that voltage-gated calcium channel on the axonal bouton. With less active voltage-gated calcium channels, you would probably expect symptoms similar to myasthenia gravis, except you would expect those symptoms to get better with use. So knowing the fundamental difference between myasthenia gravis and Lambert-Eaton pathophysiology will serve you well in your classes and on the boards. The next topic that would be good to cover is botulism. So botulism is caused by botulinum toxin. Do you remember what the target of the botulinum toxin is? Well, it's actually the snare proteins that are in the axonal bouton. And by targeting those snare proteins, acetylcholine cannot be released, therefore causing the symptoms of botulism. And one more pretty high yield example to illustrate the importance of physiology is malignant hyperthermia. So malignant hyperthermia is a condition that affects some people after they go into surgery and are exposed to inhaled anesthetics. And these people have a genetic variant of a certain receptor. Do you remember what that receptor is? It's the ryanodine receptor. And their ryanodine receptor responds to inhaled anesthetic by releasing a ton of calcium, causing muscle rigidity and dangerous hyperthermia. All right, so that's all the content that I have to cover for this episode. Up next, we'll have our practice board review questions, so get ready to knock those out. So our first question is, a 32-year-old woman presents to her primary care physician's office and suspects that she is pregnant. Her first son died 2 years ago from complications of Duchenne's muscular dystrophy. She's worried that her next child may develop the disease and wants to know how likely this is to happen. Her primary care physician correctly says that the likelihood of her next child having Duchenne's muscular dystrophy is A, 0%, B, 25%, C, 50%, or D, 100%. And the correct answer is B, 25%. So the trick to this question is that assuming the mom is heterozygous, is a heterozygous carrier for this condition, and we don't know the gender of this child yet, there's a 50% chance that the child is a boy, and then there's a 50% chance that that boy is affected, because Duchenne's muscular dystrophy is an X-linked recessive condition. So when we do the math, there's a 1 in 2 chance that this child is a boy, and then there's a 1 in 2 chance that that boy will inherit the defective X chromosome carrying Duchenne's muscular dystrophy. And that comes out to 25%. Okay, so that first question was probably more applicable for the first step of your board exams. Now let's move on to a question that might be more applicable for step 1 and step 2. So a 40-year-old man presents to the emergency department with acute severe right lower quadrant abdominal pain for the past 3 hours. He's quickly diagnosed with appendicitis and sent for emergency appendectomy. During the surgery, he develops tachycardia, tachypnea, and his temperature spikes to 103 degrees Fahrenheit. What drug should be immediately administered to reverse this patient's condition? A, oxygen, B, beta blocker, C, dantrolene, D, danazol, or E, some ridiculous answer. And the answer is C, dantrolene. So the key to this question is, one, being able to recognize malignant hyperthermia. So this patient was sent in for surgery, and then they developed a really high temperature during that surgery, also tachycardia and tachypnea. Then once you've recognized that the patient has malignant hyperthermia, you also need to know a drug that we can use to treat it, and that drug is dantrolene. So dantrolene is an antagonist of the ryanodine receptor, so it makes sense that we would be able to use it to reverse malignant hyperthermia, because the underlying cause is a defective ryanodine receptor. And with that, that's the end of this episode. So I hope that you enjoyed it. I hope that you learned something. I'm planning to be back in a week with my next episode. I'm basically shooting for an episode a week, and I'm going to do my very best to achieve that goal. Thanks again for listening to the Med School Phys podcast. If you like what you're hearing, please consider subscribing to the podcast, which is quick, easy, and free to do. Also tell your friends about it. See if this format works for them. New episodes will be available via iTunes and Stitcher. If you have questions or constructive feedback for me, you can email me at medschoolphys@gmail.com, all one word. Lastly, I should mention that this podcast is solely for educational purposes, particularly to aid medical students in their studies. It is not intended to serve as a substitute for the consultation, diagnosis, and/or medical treatment of a qualified healthcare provider. Please consult your healthcare provider regarding personal medical decisions. Thank you.